Hello everyone, this is Dr. Geetashamnani. Today I will start with one of the practical of the hematology lab that is very much useful for the first year MBBS students, and that practical is hemoglobin estimation. Hemoglobin estimation in most of the colleges in the first year MBBS students is done by the Sahli's method. So before doing the practical we must know what is the apparatus and what are the different parts of the apparatus and what are the other materials which are used in this practical. So let us see in Sahli's hemoglobinometer or the Sahli's apparatus, that is also known as Sahli's Adams hemoglobinometer or hemometer. So what are the parts? Comparator, hemoglobin tube, hemoglobin pipette, stirrer and Pasteur's pipette. So I'll show you one by one. This is the first part of the Sahli's hemoglobinometer, that is comparator. It is the rectangular box which contains, you can see these are two plates which are golden brown. These are non-fading standardized plates, color plates. So you can see these are two plates and there is a gap or slot for the hemoglobin tube. So I will place the hemoglobin tube in this slot. So here it is visible. Behind that you can see there is a rectangular white plate that could be of glass or that could be of plastic. So it is there to provide uniform illumination for color matching. So this is first part, that is comparator. Here it is written over there. Many times it is there in the Viva that students may not be able to remember the name of the method. So here it is written Sahli's method. Okay. Second part of this apparatus is hemoglobin tube. So here you can see this is a glass tube which has round or a square base and there is marking on two sides. Here it is marking with the yellow color and here it is the marking with the red color. With the yellow color there is marking from 2 to 24. With the red color marking is there from 20 to 160. But for us yellow color marking is useful because we are going to detect the hemoglobin in gram percentage. So yellow color is for gram percentage and red color is for percentage hemoglobin. Okay, so this is about the hemoglobin tube. Now next part of the apparatus is this hemoglobin pipette. Can you see this pipette? This is glass pipette. Okay, and this is a plastic tube and this is a mouthpiece, plastic mouthpiece. In the glass tube there is one marking. You can see there is one marking which is at 20 microliters or 20 cubic milliliters or 0.2 ml. Okay, so this marking is equal to 20 cubic millimeter volume. Okay, and there is no bulb in this pipette. Rest of the two pipettes in the hematology lab are WBC and RBC pipettes in which there is a bulb at the center. Here we don't have any bulb in this tube, and this tube measures the exact volume, not the arbitrary volume which are measured by the WBC and RBC pipettes. So this is the hemoglobin pipette. Now there should be, next part is Pasteur's pipette. Pasteur's pipette, we don't have Pasteur's pipette here. So in place of Pasteur's pipette one can use the glass pipette with rubber tip. In place of glass pipette we can also use this plastic dropper. Simple plastic dropper can be used in place of glass dropper or Pasteur's pipette. One more part is there, that is stirrer. So mostly stirrer is made up of glass rod, but here we are provided with the plastic stirrer, but mostly you would have a glass rod in the form of stirrer. So what is the function of the stirrer? Stirrer is used for mixing the solution. Okay, so you can see this is a stirrer. Mostly it is of glass rods, transparent glass rods, but here We are provided with the plastic rods. Now one more thing is provided with the instrument is this brush. So this brush is used to clean the hemoglobin tube. One more thing is there. Here we have N by 10 HCl. Okay, so this is desi normal HCl or N by 10 HCl, which is used for converting the hemoglobin into the acid hematin, and we are getting the deep golden brown color of the acid hematin in this practical, which is done with the help of N by 10 HCl or diluted HCl. Then there is distilled water. Distilled water is used for the dilution. And then there should be pricking material by which we are going to prick. So there may be needle or lancet, there may be gauze piece or cotton piece, and there may be spirit or 70% alcohol. Now I will show you how to do this experiment. Okay, so first we have to fill this hemoglobin tube with N by 10 HCl, and we have to fill up to mark 3 mark of yellow marking. So here I have filled this hemoglobin tube up to mark 3, and then I will place the hemoglobin tube into the slot which is there in the rectangular box or comparator. Then okay, now I am supposed to prick the finger. So what are the precautions you have to take before pricking the finger? You have to sterilize the finger. Which finger you will use? You will use either the ring or middle finger. Why not index and little finger? Because palmar fascia of these fingers are connected with the forearm. So in case, rarely, if infection occurs due to the pricking, that infection will not spread to the forearm if we are pricking the middle or ring finger. So we have to prick at the tip of the finger. So for that, first we have to sterilize the finger with the cotton swab, and that is there is spirit in the cotton swab, and I am sterilizing the finger from center to the periphery, and then give some time for the sterilization to occur. Why we need to make this finger dry before pricking? Why? Because if we are not giving time to get it dry and we are pricking and blood comes out, the blood will hemolyze. And second thing, because we need a proper drop of the blood. If there is wet, if the finger is wet, that drop will not be formed and blood will spread all around. Okay, and one more thing, blood will be diluted. Okay, and then we have to prick. Here I am pricking with the lancet. So pricking should be done with one blow only. We don't have to prick again and again. And second thing, we don't have to squeeze our finger. Why we don't have to squeeze? Because if we are going to squeeze our finger, then tissue fluid will also come with the blood and the blood will get diluted. Okay, so you prick with one blow. So here you can see you have to form a complete big drop of the blood, and before that you have to wipe out 1 or 2 drops of the blood, and now you have to dip the tip of the pipette completely inside the drop, and then the blood will so like this you have to fill the blood up to the mark 20. And then you have to pour this blood inside the hemoglobin tube, in which some amount of the N by 10 HCl is already there, and you will just push 2-3 times so that the blood is completely filled inside the hemoglobin tube. Slowly you bring out this hemoglobin pipette out of this hemoglobin tube, and then you will mix with the help of this stirrer. You will mix the solution, and then you have to wait for 6 to 8 minutes. Now why you have to wait for 6 to 8 minutes? Because hemoglobin which is present in the solution will be converted by the N/10 HCl into the acid hematin, and this will take about 6 to 8 minutes. Suppose if we are not giving time 6 to 8 minutes and we are ending this in 3 to 4 minutes, maybe all of the hemoglobin is not converted into the acid hematin. So after waiting 6 to 8 minutes, what you are going to do? You will add this distilled water one by one drop. Okay. So so here you can see the color of the solution is deep golden brown. Okay. And this thing is important, that you don't have to bring this stirrer out of the hemoglobin tube in any part of the experiment after this. Okay, this will be taken out only after this experiment is over. Okay, so what we have to do after 6 to 8 minutes, we'll just fill the dropper. Where is the dropper? We'll fill the dropper with the distilled water, and we pour one drop of distilled water inside the tube through the walls. Okay, I have poured 2-3 drops, and now I'll see the color. I'll mix it with the help of stirrer. You just have to rotate the stirrer like this, not like this, otherwise the base may be broken. So rotate the stirrer like this. You have to gently rotate the solution, otherwise the frothing may be formed. So frothing may interfere with the reading. Okay, so here I am looking for the color matching. So you have to look for the color matching against the natural light. It is very deep, so I am adding more distilled water. Now one question is there, can you add tap water in place of distilled water? No, because tap water contains impurities and that may interfere with the color matching. So can we use N/10 HCl in place of distilled water? Yes, we can use, because N/10 HCl, because all of the hemoglobin has already converted into the acid hematin. So no more hemoglobin is there to be converted. So that will not affect the this conversion. But because color of the N/10 HCl is transparent, so it can be used for dilution in case if distilled water is not available. So here I can see it is still dark. So here it is little bit dark. I have to read the reading. Marking is at about 12. So at 12 I have taken the lower meniscus of the fluid. So at 12 the solution was little dark. So this is the first reading, 12 gram per deciliter. The solution was little dark. And then again I have poured few drops, mixed it. I will elevate the stirrer against the natural light. It is matching, the color is more or less matching. Now I'll read this. This is about 12.2. So at about 12.2, second reading, 12.2 gram percent. This is matching, but I need to pour one more drop, 1 or 2 drops. Notice this point, I am not taking out the stirrer out of the hemoglobin tube, I am just elevating it. So here it is little lighter. It is 12.4. So third reading is 12.4. Now there is some controversy. Some of the colleges are considering that you have to take average of 3 readings, and some other colleges are considering that reading before the last one, means when the solution has diluted, the reading before the diluted reading is the true value of hemoglobin. So whatever your college is believing, you You just keep that criteria. If your college is telling you the average, you just do the average of 3 readings. I strongly believe that we should take reading just before the dilution, because many times it happens that matching does not occur at one point. It is possible that matching is there at 12.2 and matching is there at 12.4, but it is become diluted or faded at 12.6. So in that case, what will you do? You will take average? No, you will take just the reading before the diluted one. Okay, 12.4. So this is about the hemoglobin estimation. Now there is a possibility that the hemoglobin of the person you are performing is normal, but due to some error that hemoglobin comes out to be low or high. So what could be those errors? So now first discuss the error which could lead to the decreased level of the hemoglobin. So where could be the error? First of all, when you are pricking, in case if you are squeezing the finger, plasma comes out. Okay, that plasma mixes with the blood and leads to the dilution of the blood. Suppose if there is sweating at the fingertips, or you are not giving time for the spirit to dry, so blood will mix with it and it will become diluted and it will give falsely low value. Then what could be the possibility when you are filling the hemoglobin pipette? Okay, during filling the hemoglobin pipette, maybe there is a possibility then there are some air gaps, so amount of the blood will be less than the 20 microliters. Then during pouring that blood, you are not completely washing that blood inside the hemoglobin tube, and some of the blood is sticked in the walls of the hemoglobin pipette, so that blood is taken out of the hemoglobin tube. Okay, so that will also give you falsely low value of the hemoglobin. Next, if you are not mixing the solution properly, if the solution is not mixed properly, then also it will give the low value. Then there is a possibility that you are taking out stirrer each time. Okay, so even if the micro drop of the blood is taken out through the stirrer, it will give you falsely low value of the hemoglobin. And then if you are not reading it properly, okay, if you are not matching it properly, that may also give you falsely low value. Thank you. Now you must know what could be the technical errors which can lead to falsely high value of the hemoglobin. So first of all, there is a possibility that in the pipette you have taken the blood more than 20 microliters, or there is a possibility that when you are taking blood at around the tip of this pipette, because this tip is completely inside the drop, you have taken some blood around the tip. So if that will go inside the solution, that will give you falsely high value. So you have to wipe the tip of the pipette before pouring into the hemoglobin tube. Okay, but you should not put cotton at the tip, otherwise it will soak the solution inside the pipette. Okay, so then you have to do it. In case if you are doing excessive mixing like this, okay, so that will lead to frothing of the solution. In case if there is frothing, it will give you falsely high level of the hemoglobin. Again, if the solution is over diluted, so that will also give you high value. And if in case, also rarely it is possible that the glass plates are faded, okay, although they must be non-faded and standardized, but there is a possibility that after decades these glass plates are faded. So in that case, it will give you falsely high level of the hemoglobin. Now you must know what are the common viva questions in this practical. So the first question your examiner is going to ask you, what is the normal value of hemoglobin in men and women? So in men, the normal value is considered to be 13.5 to 18 gram per deciliter, and in women the value is from 11.5 to 16 gram per deciliter. So this is normal value of hemoglobin. Now your examiner may ask, can we use strong H2SO4 or strong HNO3 or strong alkali in place of HCl and by 10 HCl? So the answer is no, because these strong acids are going to disrupt the hemoglobin. they are going to destroy the hemoglobin. So we cannot use these strong acids or alkalis. Weak alkalis also, weak acids and alkalis also we cannot use, because this experiment was standardized taking the N/10 HCl. Okay. Now the next question of your examiner may be, because we are going to use N/10 HCl. So what is the meaning of N or normality? What is the meaning of N? So it is the gram equivalent weight of that substance in 1 liter of the solution. Suppose for the HCl it is 36 grams. H, 1 gram for H and 35 for Cl. So 36 grams of the HCl in 1 liter of solution is considered to be the normality 1. And if we are diluting it 10 times, it is N/10 or deci normal. Next question of your examiner may be, should you take out the stirrer every time after mixing? The answer is no, as I have already explained, because it may take out micro drops of the blood each time, and that will give you falsely low value. Then your examiner may ask you what will happen if too low amount of the HCl, N/10 HCl, or too high amount of the N/10 HCl is taken in the experiment. Then your answer must be that if HCl is very low, in the beginning, if N/10 HCl is very low, it will not completely convert the hemoglobin into the acid hematin. So it should not be taken. It should be taken up to 3 marks only. In case if it is taken more than that, suppose if you are taken up to 4, 5, maybe up to 6. So if there is a possibility that the actual hemoglobin of your patient is 5 and you are already taking HCl up to the 6th mark, then what will happen? In the beginning also the solution will become diluted and you will not be able to give the correct reading of hemoglobin. Okay, the next question may be that if what will happen if you are not giving 6 to 8 minutes, maybe you are giving 2 to 3 minutes, what will happen? The hemoglobin will not completely convert into the acid hematin if the time given is less. If it is much more, like if you are giving 1 hour or 2 hours, then the color of the solution will be faded. Okay, then your examiner may ask you what is the oxygen carrying capacity of your blood. For that you must know that 1 gram of the hemoglobin carries 1.34 ml of the oxygen. 1 gram of hemoglobin carries 1.34 ml of oxygen. So if the hemoglobin is 13 grams per deciliter, so it will carry 13 into 1.34. That will come out to be 17.42 ml of oxygen per deciliter of blood. So this much ml of the oxygen is carried by per deciliter of the blood if you know the hemoglobin concentration of any person. So this was all about the hemoglobin estimation and the common viva questions related to the hemoglobin estimation. Thank you.